Hello, my name is Sana Badri, and today I'll be discussing a project I've been working on at the NYU School of Medicine studying TF gene regulatory interactions to identify new therapeutic targets in CRLF2 overexpressing BALL. Significance of characterizing the underlying molecular profile for each patient is useful for the development of personalized therapies and can inhibit specific targets in the cell and lead to less damage to healthy cells. And so a lot of times cancers are defined by various subtypes according to specific genetic alterations. Here we are interested in studying alterations involving the CRLF2 gene in B-cell acute lymphoblastic leukemia. And CRLF2 is a cytokine receptor. It's also known as the TSLP-R receptor. And the reason why we're interested in it is because it's associated with high-risk BALL and poor outcome. And this alteration can occur either due to chromosomal translocations or activating mutations. So the function of CRLF2 is that it binds the TSLP protein and then induces the phosphorylation of STAT5, PI3K, and ERK MAPK pathways. And there are available inhibitors that target these pathways. And there have been studies that evaluated the use of these single inhibitors in this context, but have resulted in limited efficacy. More recent studies have looked at combining these different drugs and have shown improved efficacy, but there still needs to be more work done to evaluate the toxic effects and exactly the biochemical mechanism of using multiple drugs to target these cells. And so our goal here was to better understand the impact of CRLF2 overexpression, to better understand gene regulation in this context. And so we wanted to construct a BALL-specific regulatory network to define the regulators of the CRLF2 high gene signature. And so the first thing we did was identify differentially expressed gene signature between CRLF2 high patients that had high levels of CRLF2 and other BALL primary patients that had 0 to minimal levels of CRLF2. We also generated a transcriptome, BALL transcriptome from hundreds of primary patient RNA-seq samples from the target database. We did this to be able to generate a BALL-specific regulatory network and constrain this network using attack motif-based priors to basically define these regulatory interactions. And so we then leverage those interactions to see what are the regulators that are important in regulating the genes affected by CRLF2. So transcriptional regulatory networks allow us to better understand the relationship between TFs and their gene targets. And we can visualize these networks as these kind of various nodes where you have source nodes as TFs and target nodes as gene targets, and they're connected by these edges that oftentimes have a sign associated to them. And typically the sign can either indicate activation or repression, and we can infer these networks using either expression data alone, or we can combine expression data with multiple data types. And so the limitation of using just expression data is that there are a factorial number of possibilities, and TF transcriptional expression levels are not a good proxy for the transcription factors activity because most TFs are post-transcriptionally controlled. And so we try to estimate the activity. And so to give you an example of why this is important for inference, so you can imagine if you have a TF and the region where it's supposed to bind, it's inaccessible or it has closed chromatin, then that TF is not going to be localized, binding and acting and driving expression. And so it's important to incorporate different data types like attack-seq data to get a better idea of what's happening in the cell and to lead to more accurate inferences. And so what is transcription factor activity? We define it as the relative activity of a protein that is inducing or repressing transcription of its targets in a sample. And we can estimate TFA based on the partial prior knowledge of TF's gene target. And so in this case, we have gene expression from target, and all we need is basically a list of putative interactions between TFs and genes, and we're able to estimate activity. And so to do so, we need this prior in this context, this prior matrix. And so we built our priors from patient-specific attack-seq data, where we first define kind of a landscape of all attack-seq peaks across CRLF2 high and other BALL patient samples. We then search for known TF motifs at all attack-seq peaks within a 20 kb window upstream of each gene's TSS, and we then aggregate those TF motifs and assign an enrichment score of that TF found at that particular gene to generate this prior matrix. And so with that, we're able to assume gene expression can be modeled as a weighted sum of activities. Using those estimated activity levels and the gene expression matrix, we can then solve for the beta coefficients or the regression coefficients, and these represent the magnitude and direction of each regulatory interaction. So you're going to have a list of betas that define that kind of regulatory interaction, between each TF and its gene target. And so we apply this to the BALL data set, and we're able to define a patient-specific BALL regulatory network. And when we filter for high confidence interactions, we're left with about 8,731 regulatory interactions between 135 TFs and 6,645 unique gene targets. And so we then want to go in and determine what TFs within this network could possibly be altered at the activity level as a result of this genetic mutation. And so what we thought to do was to use the initial data set that we had applied to generate the CRLF2 high gene signature, which was also excluded from the inference process. And what we wanted to do was see if we could then estimate activity just in these samples, but using the newly inferred regulatory interactions. So all we needed was the new inferred interactions, the expression matrix for these 30 patient samples, and we're then able to generate a matrix of estimated activity levels for about 135 TFs. And so then we can interrogate which TFs are potentially altered or have a differential mean activity level. And so we are left with about 36 potentially significant TF regulators that are altered at the activity level, which we then subset the network for these altered TFs and their differentially expressed gene targets, and we're left with this CRLF2 associated subnetwork of 34 TFs and 542 gene targets. We further want to dig deeper into this subnetwork and see if we can filter this list a little bit more by looking at the most robust gene targets that could be the most likely targets to evaluate for therapy. And so we did this by using a different model where we used cell lines, 3 individual cell lines, 2 of which have high levels of CRLF2, Call and Mutz, and SMS, which has low levels or non-existent levels of CRLF2. And we then compared this with the patient samples that we had, and we defined 67 concordant differentially expressed gene targets across patients and cell lines. And so at this stage, we have identified a list of putative targets, but we want to validate whether our approach and our predictions could be useful. And to do so, we thought to analyze public experiments in related cells to validate some of our predictions. And we were able to find about 9 experiments, either ChIP-seq, siRNA, or CRISPR-I experiments for about 9 TFs in our subnetwork that we could use to analyze to validate some of our predictions. And so here you can see the results of this analysis, where we have validated a proportion of the inferred gene targets for each of these 9 TFs. And we then went in to see whether this proportion was statistically significant compared to random chance. And for the most of them, with the exception of the E2S2 transcription factor, they are statistically significant, but we expect that if we had looked at the context where CRLF2 is overexpressed, that we could potentially have even more validated gene targets. And so to summarize, we have inferred transcriptional regulatory interactions downstream of CRLF2 mediated signaling in BALL. We have estimated TFA of TFs in CRLF2 high in other BALL patients, and we have identified a list of significant TF regulators and their differentially expressed gene targets that were not previously known to be associated with CRLF2 overexpression. Furthermore, a number of these regulators and their gene targets have been implicated in important processes such as self-proliferation and survival, drug resistance, immune response, as well as many more that could be giving these leukemic cells an advantage. And so with that, I'd like to conclude and thank my two labs, the Skok and Benel lab, as well as our collaborators, the Carroll lab, the HPC facility at NYU, and thank you to everyone that was here.